Hello, this is Dr. Paul Cottrell, and I'm going to be talking about a group of researchers in China that were doing cancer research, and they ended up contracting the cancer that they were studying. And this is related to two paths of investigation that they were doing. Now, again, all this is in Chinese, and I'm going to show you some slides and stuff, so bear with me. But it seems to me, without having an English version of this, it seems to me that there was a line of research in terms of making cancer cells or turning cells into cancer. And another line of research on how to use antigens, cancer antigens, as part of a therapy, an immune therapy, to fight those cancers. So let me restate that. It seems to me that these researchers that I'm going to discuss were manipulating cells. It looks like it was in vitro, but potentially in vivo studies also. But manipulating tissue where they were taking healthy tissue and turning it into cancer, and using immune therapy to be able to fight that cancer. Okay, so let's dive in. This is information that was given to me from Miss Taiwan back in November of last year, of 2023. This is the reason why I'm doing it now, is that now I have time to kind of dive into what she was sending me. Now, there were 6 doctors that are from China that ended up getting cancer. They were research doctors, all right? And they were focused on studying a certain type of cancer. There was a cover-up that took place in China, and one of the researchers that ended up getting the cancer was trying to be a whistleblower, and her story was covered up. Now, again, it looks like the researchers were developing methods to create cells or tissue that turns cancerous faster, and also a treatment for that. Okay. Doctors were in their 30s, and many of them had developed pancreatic cancer. Okay. The woman that was the whistleblower was a female doctor. Her name is Hyung Ming. So I'm gonna spell it Huang, and the last name is Meng. Female doctor. And it was last September when of 2023, where she was having pain. So most likely back pain and shoulder pain, right? And they did a workup, and she had higher levels of pro-inflammatory cytokines. So there was another examination that took place in October that staged the cancer for pancreatic cancer. So probably blood work and radiological work, biopsy, and all this. So she ended up getting stage 4 pancreatic cancer in her 30s. Looks like they were manipulating a certain gene that is actually a tumor suppressor gene. Right? It's called SMARCB1. Its alias is INI1. So just for simplicity, I'm just going to refer to INI1. Now, why is this important? That gene, when it creates its protein, that gene, when transcribed and translated, is factors that help to release the DNA from the histone, unravel it to allow for gene expression. And those genes happen to be tumor suppressor genes. So if INI1 I1 isn't working properly, then you are suppressing tumor suppressor genes. So you'll have more cancers. What is interesting about this is that when there's a mutation on INI1, cancers start to happen. But it looks like that if INI1 is in its normal form, non-mutated form, and in its mutated form, that it helps with integrase for HIV. What that means is that if someone had HIV, that it helps with integrating that retrovirus into cells genome. With a retrovirus, it has to go through a reverse transcription plus an integrase to be able to get into the cell. It happens to be that INI-1 helps with that process. It helps that to allow for the DNA to bind together through this HIV-1 integrase. Now, throughout this whole coverage of this crisis, in the post-crisis, HIV is always in the background. So she tried to share the news, but it was being buried by it. Now, the director, now what I'm going to do is I'm going to show you the actual report that the researcher, this is her lab report. This is the report of Kyung Ming, the doctor that ended up getting the pancreatic cancer. And I'm going to zoom into one area, and this is the area we want to talk about, that there's a mutation that's taking place with the SMARCB1 and the IN, or the alias, the INI1. So this is really important to kind of pay attention to. Now, okay. Now, what she was working under a director, research director. The name of that research director, I believe the director is female, but I may be wrong. Maybe male. Su-Shi Cheng. So I'm going to spell it S-U, then S-H-I is the second part of the name, and then the last name is C-H-E-N-G. Now, this researcher is a director, and it happens to be a member of the CCP. Well, I think a lot of people in China, if they're moving up the ladder, are part of the Communist Party. So I don't see that as a big different, big revelation. But her focus, the director's focus, Dr. Su, is her focus was on breast cancer and immunology. This is important because remember what I said. They were finding ways to accelerate cancer, and they were finding ways for the treatment of that accelerated cancer through immunology. All right. So this person that was trying to blow the whistle that there was something going on at the laboratory, that 6 researchers in a very short amount of time ended up getting turbo cancer, all right, was suppressed in the mainstream media in their country, in China. So there was this WeChat that was going on, and someone picked up that WeChat and started compiling this information that I'm showing you. So, and then Miss Taiwan was watching that person's compilation. Now, 5 other doctors in the research also ended up getting cancer. One of those doctors is a male that ended up getting breast cancer. So my understanding is there's 3 cancers within that 6-person group. That's pancreatic cancer, lung cancer, and breast cancer. Now, why is that important? It's important because if you knock out or mutate the IN1-1, not only does a normal IN I-1 help to unravel the DNA so it can transcribe tumor suppressor genes, but it also works in tandem with a bunch of other tumor suppressors. And some of those are BRCA1, breast cancer, MYC, which is heavily involved in cancer, P53, cyclin-dependent kinase 8. So, the point I'm making here is that by mutating or manipulating INI1, then you're also affecting some other pathways that can be problematic for cancer development. So there was this suppression that was taking place, all right? But there's these other doctors that in a very short amount of time contracted it. Now the question is, how did they contract it? Now, did they contract it through the manipulation of the cell lines to get them to be more cancerous, or did they get it through the immune therapy? Now, the Chinese hospital posted a notice, and I don't know what the Chinese hospital is, admitting that these doctors were associated with breast cancer research and that there were 200 patients or something there's tied to the study. All right. So but I don't have a paper to corroborate that. And of course it's in Chinese, so I don't know. But the research was closed down. All right. And there's been very little talk about this. So it's been hushed. Now, Dr. Hume, the one that has the pancreatic cancer that was trying to blow the lid, I showed you the INI-1 report showing that she had some sort of mutation. Now, my take on this is, now I want to show you the director, Dr. Sue. Let me show you that picture. Okay. This is the director. Now, honestly, I can't tell if that is a male or female. It might be a male. But that's the director that shut down the program. Specialty is in immunology and breast cancer. And the research that these 6 doctors working under this director were doing was causing mutation in such a way that the cancers were manipulated to be more aggressive or to start to develop, and that they were coming up with some sort of treatment. Now, why do I say that they were coming up with some sort of treatment and it's tied to immunology? Because this here is part of the paper, but I can't find it, right? Because it's all in Chinese, and so there is this piece of a paper, all right, talking about what they were doing on the immunological side of it. I'm going to zoom into the graph, okay? And that's here. Now this graph, all right, is an immunological graph. Now I'll walk you through it. So at the very bottom is a tumor, okay? And that tumor is producing antigens, all right, cancer antigens. And that APCs can start grabbing onto those antigens and starting to present them to the lymph node. And when they present them to the lymph node, then T cells and B cells start to become activated. Now, this chart is focused on the T cell. Now, if you want to understand some of the dynamics of this activation in the paracortex and the cortex of the lymph node, and what's going on with that communication and some of the cytokines and some of the signalings, I have a video that goes into detail. I think it's white lung, why it's happening, part 2 and part 3. So please go to that video to get that understanding for this section, the priming and activation section. Then once the T cells get into the bloodstream that have been activated, primed and activated, then what happens is that they're designed to pick up, to attach to cells that have this antigen. Okay, so it'll get through the lining of the blood vessel and get into the tumor. Okay, and then the tumors that have the cells within the tumor that has those antigens that B cell is designed to attach to will attach and then create a killing mechanism to kill off the cancer. Now, in a tumor, there are many things that are going on. Now, I'm going to be roughly generic with my explanation here because there's a lot of nuance and there's a lot of detail, right? But cancer cells start to divide, and then as time goes on, there's more instability in the genome of the tumor, and so there's like different genomic populations. Okay, and some tumors may be presenting some types of antigens at a higher concentration than others because of this dynamic. And there are a lot of cancer antigens that are probably in that tumor, not just one type. So the lymph node in the priming and the activation stage is producing a lot of different type of T cells to focus on one particular antigen. So you may have a population of T cells that attacks different types of cancer antigens, all right? And then create their killing mechanism. Now, that is the way it's supposed to work, okay? Now, it looks to me that the research they were doing were trying to speed up the process of producing cancer in the laboratory, probably to speed up the laboratory time to get results, but also to try to understand some of the mechanisms that are going on in the cell. And it looks to me that they either manipulated the IN1-1 pathway to do that, or they did some other pathway, and that their immune therapy for this ended up creating turbo cancer. Okay, so let me reiterate here. Okay, so now you understand there were two pieces to this research that was going on. Manipulation of some pathway to advance cancer or produce cancer, to help with understanding those pathways and potentially speeding up the process for results. And then there was another leg of their research that was dealing with therapies for breast cancer using immune therapy. All right. Now, that immune therapy could have been monoclonals for a particular antigen, or it could have been inoculation through some sort of vector, right? to try to elicit an immune response to build up T cells and B cells against the tumor, right? And we've heard this being investigated and developed by, the individuals that are household names now in these companies are household names since the crisis. I don't want to say it because I want to keep the video up. But the point here is that it looks as though these researchers were doing two things: accelerating cancer through manipulation of pathways, potentially INI-1, and or the therapy ended up mutating IN1-1. Okay. Now, what do we know from this information? There were 6 doctors in a research study that ends up getting cancers at the same time, 3 different types. Most of them were pancreatic. One was breast cancer. The other, I believe, was lung cancer. Okay. I think it was one that was lung cancer. But there were 3 different types of cancers. They were playing around. What we do know is that from the report, the lab report, that mutation of INI-1 for the pancreatic patient. We don't know the reports for the other 5. We do know that they shut down the research. At least they say. The point I'm making is that they had research that shows terminal cancer being developed in one of 2 ways. One, through the mutation of the INI-1 pathway, and or immune therapy that ended up causing problems that led to the terminal cancer. All right. So I think this is another data point that shows that at least the method of immune therapy that they were doing adds to the chances of terminal cancer. Now, there's a couple things that bother me about this. All right, and I'm going to walk it through with my line of thinking here. The researchers, all 6 researchers, ended up getting cancer, advanced stages of cancer. That sounds like terminal cancer, all right, in layman terms, okay? And we don't know the method of how they accelerated the cancers in that leg of their research study or studies. We do know that they were using immune therapy. Now, it is possible that for that pancreatic patient, the researcher that ended up getting pancreatic cancer, it's possible. We do know that they have a mutated IN1 pathway. The question is, how did the 6 researchers get the terminal cancer? They were researching terminal cancer. They were researching therapies for terminal cancer. It's possible that the therapy produced the terminal cancer. All right. They were researching how to accelerate cancer, and they were researching how to treat it. And the treatment could have helped with the terminal, is my point here. How did the researchers that were not participants in the study, they were the researchers of the study, get it? There's the big question. I don't know the method of how they accelerated the cancer before the treatment. All right. And I'm not sure if it was in vivo studies or in vitro studies. All right. But they had, it seems as though there was 200, I don't know how many, I don't know how many samples they had, or how, if there were patients that were involved or animals involved or what. I don't know. So, and it's, I think in the facility there were around 200 students. So, and that's, I think they informed the students that there was a problem. So the issue that I have is how many people were actually in the study, patients, or how many animals were in the study, and how much of this was in vivo and how much of this was in vitro. And what was the mechanism that they used to accelerate the cancer? Was it a carcinogen, a chemical, and that these researchers were exposed to that chemical because of improper laboratory procedures? Or was it that they used some sort of transfection method and recombination method to mutate the cell lines or the animals to accelerate the cancer, and somehow that transfection, either through viral-like particles or through some sort of viral vector that they were using, ended up getting into their body? I doubt that all 6 would have had an accidental needle poke. I doubt that for all 6. 1 maybe, but all 6 no. Maybe it's something that is airborne, and they were only working with, let's say, a BSL-2, a P2 lab, and they weren't properly protected for it, and it ended up giving them cancer. Possible. That's on manipulating tissues or animals or whatever to accelerate the cancer. And or it could have been the treatment, where they were administering, either in vivo or in vitro, some sort of viral vector or bacterial vector or bacterial phage or a virus-like particle, whatever, to help with the treatment. Because I suspect, because it's an immune treatment, that they used in vivo study, probably with animals, to test the immune therapy, and that somehow, some way, they ended up getting it. Now, was it through some sort of nasal spray? Was it, improper procedures that ended up going airborne? And for all 6 of these, it's very odd. So just kind of like sit back and go, there are more questions than answers to this. It just seems as though there have been research that's going on that shows that here's another data point that we have cancer, increases in cancer, an accelerated cancer. And there was a study that was going on in China for breast cancer that ended up giving the researchers cancer. Now, that could only happen if they were exposed to the same carcinogen or if they were exposed to the same viral particles or whatever. I don't think it's some sort of I don't think it's the typical chemical carcinogen that you would, you would think, like benzene or something like that, right? I suspect that it has something to do with like nasal administration, nasal spray administration, or dealing with viruses that ended up going airborne somehow that they didn't think would, and they had a poor safety protocol for all this. And they were dealing with something that was more infectious than they thought. Again, I don't have the answers, but I just want to, it seems because I don't have the paper of what they were publishing, it's hard for me to dissect the information that Miss Taiwan gave me from this hodgepodge WeChat that was going on with this doctor that had the pancreatitis. The person that did the WeChat was the doctor that had the pancreatitis. Not pancreatitis, the pancreatic cancer. So the point that I'm making here is that there's a lot of unknown that other people can maybe dissect and figure it out. But it looks as though in November there was discussions that were going on in WeChat of this doctor trying to blow the whistle on this research that was going on, and these 6 doctors ended up getting cancer. Now, did they get the cancer because of the accelerated protocol that they used, or was it that they ended up getting the cancer because of the treatment? We don't know. I suspect it's the treatment. And I suspect that some of the similar things that we're starting to notice with turbo cancer in the post era that we're in, the post-crisis era. I'm being generic for this, trying to keep this video up. We're seeing something very similar. These turbo cancers that are popping up. So now, why is this so important? Well, the easy answer is, well, if you're studying cancer and you're trying to create a therapy, then it's for scientific discovery and medical knowledge, right? The other side of this is this weapon system, where more knowledge is being gained to be able to manipulate certain types of tumor suppressor pathways. In this case, it looks like it's the INI-1 pathway. All right. Why is that important? It's important because it helps with one, HIV-1 integrase. And in addition, if INI-1 is mutated, you are suppressing the tumor suppressors, leading to certain types of cancers. And there's these interplays with other tumor suppressors like P53 that coincide with the INI-1. So there's more questions than answers. I don't have the answers to these questions. It goes dark because the research stopped, supposedly. And not only that, all this stuff's in Chinese. So it's not very easy to kind of like understand exactly what's happening, but that's the best that I have to show. Now, potentially some tangential data points are what I'm going to show you. Now, the director Sue, right? This director here, this director, male, female, I don't know. This director, probably male. This director had a child. All right. And that child was a high school student. All right. Was a high school student and was put on a paper. Now it's probably, give the kid, some sort of something to type up in a research study and blah, blah, blah. But what's important is the other people that are on this list. They're dealing with Institute of Cancer and Stem Cell, right? In China, different types of medical facilities. There's a pathology department. So there are people here that may be able to triangulate on this research study to help get closer to understanding what they were doing with the immune therapy and the manipulation of the IN1-1 pathway. Now, what's interesting on this research study is that they're dealing with the nasopharyngeal carcinoma. All right. And like I said, were they playing around with some sort of nasal spray? And here, the macrophages, this is the whole immune therapy. Here again, nasopharyngeal carcinoma and the activation of macrophages. Now, I'm sure that this kid didn't do much in terms of contribution to the study because his parent, was tied to major research in cancer. So this was probably just a resume booster for them to get into medical school or whatever. What's more important is who else is on this list. And it so happens to be the parent, which is number 6. Number 6, Provincial Key Laboratory of Malignant Tumor Epigenetics and Gene Regulation. So there's some data points here that can be investigated to try to understand what was going on. Now, in this one, I don't know if the mother or I don't know if the parent was on the list. I don't think so. So those are 2 data points that might get us to understand. These were research studies that were published in 2017 and 2018, right? So the contribution of the high school student, I don't think is much. Maybe they were just the lab rat, running around cleaning test tubes or something. I don't know. But the thing is that there are people that are tied to the cancer research in China that seem to be tied to the parent, which is number 6 here, Shi Ching Su. And get a better idea of what was going on. That's really all I know about this director. All right. And that's pretty much all I know about their research. My understanding in conclusion here is that it looks like that they had 2 legs on the research. One is how to advance cancer to grow and how to treat it. And they were using immune therapy to treat it. And it looked like they were doing kind of like an inoculation method. Maybe they were doing monoclonals. I don't know. But somehow, some way, 6 of those researchers ended up getting turbo cancer for pancreatic cancer, lung cancer, and breast cancer for a male breast cancer. All right. So something's weird. And I suspect that the immune therapy is what drove the turbo cancer. That's my gut feeling on this. But that's it. That's all I know about it. So hopefully you've learned something a little bit more about, you can manipulate stuff in the laboratory and things can go wrong and researchers can get hurt. Now, usually, it's like breathing in fumes and you get some sort of chemical burn or something like that, something in your eye, or you get poked, and you might get some sort of disease from being poked because, you're using all these syringes, especially with animal models and all this. But it's very odd that all 6 of those researchers ended up getting turbo cancer. At least that's what's being reported. And we know at least one of the 6 for sure has a mutation in the INI-1 pathway. All right. So hopefully you learned something. Please go to my store, the-studio-reykjavik.com, and I have a great toothpaste that will help to freshen your breath, whiten your teeth, reduce gingivitis, reduce gum inflammation. It does not have any fluoride in it. It is a structural nanosilver toothpaste, and it will neutralize pathogens. This is the best toothpaste that you're going to be able to buy. All right, they're right here. It's not Colgate. It's not AIM. It's not Arm & Hammer. This is the best toothpaste that you can buy. So go to my store, the-studio-reykjavik.com, and purchase a couple of these tubes, maybe 4 tubes or 2 tubes, whatever you want. But please, on a regular basis, go to the store and get your toothpaste and help support my work. It's the-studio-reykjavik.com. Link is in the description of this video and all my videos. In addition, we're going through some problems in the world, and you need to start following my protocol. This structural nano silver gel will neutralize pathogens. You put it on your hands, it stays active for 5 hours. Put it around your mouth, lightly coat your nostrils, around your nose, around your eyes, around your ears, lightly coat your ear canal, and it'll stay active for 5 hours. All right, helps to neutralize pathogens. It can also be used as a skincare product. I also have a lot of different types of structural nanosilver soaps. Very high quality structural nanosilver soaps. This one is lemongrass. And this one here is charcoal tea tree. I have 5 different versions of structural nanosilver soap, and this one's an oatmeal spice. But I have peppermint and lavender. So please go to my store, the-studio-reykjavik.com. I've partnered up with Rainbow Herbals, and we've developed an all-natural deodorant, all right, made from Himalayan essential oils. Very high quality. We have it in 2 different scents. One scent is in citrus, and the other is in peppermint, tea tree, lavender. It's for males or females, and it also helps to detoxify your body. So not only is it used as a deodorant, but it's a natural detoxifier. So this is going to be a deodorant that's actually going to help with your health and improve it. So go to my store, the-studio-reykjavik.com, and get a couple of these bars of deodorant. Again, it's for males and females. It is a very, very high quality deodorant. I have magnesium and zinc to help boost up your immune system. You should be taking this every day. If you're not feeling well, take a double dose of this. Maybe once in the morning and 1 in the evening, right? It is known to help to boost up your immune system. And we're going through a new wave that's coming, all right? And you need to start to prepare your immune system. You have time to prepare for your immune system. But, how do you do that? How do you prepare? Take things that boost up your immune system. Take things that will bring down reactive oxygen species and bring down inflammation and neutralize pathogens, right? Silver gel neutralizes pathogens. Max 35. Take a teaspoon of it a day. If you're sick, take a tablespoon or 2 a day. That will neutralize pathogens. You put it in your mouth, you swish it around, you swallow it. All right, and that's one part of the protocol. C60 is a very strong antioxidant. By being able to keep your cells healthy and to have ATP, it's going to help to keep that tissue at a higher level of performance in metabolism, and it will help to boost up your immune system. Why? Because if you have more ATP, you'll have a more robust immune system. That's part of it. If you have a low ATP, your immune system starts to go down. But the key here is this boost up your immune system. Structural nanosilver, liquids and gels, C60 to neutralize free radicals, and take turmeric and ashwagandha. That will bring down the inflammatory response, the pro-inflammatory response. You want the turmeric to bring down that inflammation, and you want the ashwagandha to control your blood glucose levels. That's the protocol. All right. Take it every day, and you're going to see an improvement on your health and boosting up of your immune system. And it's an anti-aging protocol because it's boosting up your immune system. It's slowing down that aging process because C60 is a strong antioxidant. You'll start to notice that your body's healing and that you're slowing down the aging process. Also, start preparing and make sure that you get my silver lozenges. All right, I have it in a 20 count in sweet menthol, and I also have it in green apple 20 count. Some people also like the honey and lemon drops that I have in the 100 count, and I also have it in blueberry. So please go to the store and purchase your lozenges and your drops. All right, things are going to get worse. It's not going to get better anytime soon. All right. Things are going to get worse. Be prepared. Please take my advice. Boost up your immune system and get the products so you have the tools that you need to deal with a cold or something else when it comes. All right. Ligaments. Again, it helps to boost up your immune system. It's for males and females, and it also helps with hormonal health. What did we just talk about? We talked about turbo cancer. Immune systems that are working properly will help to fight cancer. It's not a cure, but a better immune system is much better in fighting cancer than a weak immune system. So please listen to my advice. The wave in terms of hospitalizations is getting worse. I am concerned about the breaching of the 40,000 weekly new hospitalization count. There's going to be social pressure in the media for, certain types of mandates. Again, I'm trying to talk generically here. And there's going to be fear that it's going to start to be produced. And you need to just be prepared. All right. It's real. It's no way, it's not about acute deaths. It's not about that. It's about long-term latency and chronic. And that's where most of the population is going to be affected, not in deaths. It's the wrong metric. It's about the latency and the chronicness. And that is through both past natural infection and inoculation. I can't tell you enough on how important it is to start getting prepared. All right. The world is spiraling out of control, and the higher the chance of unconventional warfare is increasing. Be prepared. Listen to the protocol. I've been saying this for 4 years. Neutralize pathogens, bring down the inflammation, bring down those free radicals. Structural nanosilver, liquids and gels, C60, and ashwagandha and turmeric. And then there's other things that are important, like the D3 and the zinc and the vitamin C and the resveratrol and all this stuff. But please go to the store and get the products. Please subscribe to all my channels. I have 3 channels on YouTube. I have Brighteon, Bitchute, and Rumble. Please subscribe to those. Please share the links, especially the ones that can stay up. And please tell your social network to subscribe to all my channels, especially the YouTube channels, because they're heavily censored and YouTube brings down the subscription count. I'm not the only person that this is happening to. There's many people that this is happening to. Please help support us. We need your support to fight the censorship. Now, for the ones that have been watching me for 4 years, I've gotten you this far. I'll get you farther. Just listen and help. Thank you for paying attention and watching the channels. Please share the links. Please tell your social network to follow me. Things are going to get worse. Thank you for listening and have a nice day.